So this project is about how proteins that are present both in males and females actually work differently in both sexes, and in fact sometimes in an opposite way. So in particular, the protein I'm talking about is an RNA binding protein called UNR. In mammals it's called CSD1. It's a very conserved protein. And in males, it's involved in the activation of a process called X chromosome dosage compensation. And in females, it represses the same process. So in males it activates, in females it represses. In females, the protein represses a component of the dosage compensation complex, a protein component at the level of translation, the protein MSL2, while in males, it promotes dosage compensation complex formation by binding to a different component, the long non-coding RNA Rho-X. The work actually has happened thanks to the collaboration with two very strong German labs. In the case of females, the complex formation of UNR with sex lethal to repress the translation of MSL2. This structure that we have obtained has been obtained thanks to the lab of Michael Sadler and his postdoc Janos Hennig. In the case of males, where UNR is regulating a long non-coding RNA, what UNR we think is doing is to actually act as a kind of a chaperone, modifying the structure of this RNA so that other components of the dosage compensation complex can associate then form the complex. For this, we established a collaboration with the lab of Peter Becker, who had performed structural analysis on the Rho-X RNAs, which were the RNAs to which UNR was binding. And in the lab of Peter Becker, Sylvain Manner was the postdoc in charge of the work. And in my lab, these both works were managed by a PhD student, a very talented PhD student in my lab, Cristina Militi. And I have to say that, from my point of view, that I'm in the grading committee and I've been for many years there, I think I really like to see how students take advantage of these opportunities and how this helps them progress in their careers. Many people say, you're working on flies, this is irrelevant, you should work on a human disease and stuff. But I tell you something, working on flies had let me study human disease, It's something that has happened naturally. It has not been forced. And why? Because Drosophila and humans, at the molecular level, we are very similar anyways. With UNR, this protein that we have described in this dosage compensation job, we're now studying the role of human UNR in cancer, and we are studying this because our previous results in Drosophila let us suspect that this protein could have conserved roles, especially in human disease.